So this is, I put this up because it references your book, but this is just a description of the kinds of secretory pathways that we have. The constitutive pathway. It's nice because it shows you how proteins in the lumen of that were in the lumen will end up facing the outside world, as well as proteins that were totally in the lumen. Those again are what are going to receive the modifications. And it also shows you the regulated secretory pathway that some cells have. But what we want to talk about right now is, so those are two of the pathways from the trans-Golgi network. Now we want to talk about how proteins get from the trans-Golgi network to the lysosome, and they first get to a compartment called the late endosome, which is also called the pre-lysosomal compartment. So we're basically looking at how the trans-Golgi network proteins, how the vesicles budding from the trans-Golgi network recognize lysosomal enzymes and divert them to the late endosome so this can actually mature into a lysosome instead of being secreted to the outside world. And the story is, so what do we mean by lysosomes? Lysosomal enzymes? There's a lot of them. So these are enzymes of destruction. The lysosome is what receives vesicles from the outside world and then destroys whatever was in that vesicle. It's super important in the immune system. So neutrophils and macrophages, you would die without these cells, and they work because they are full of lysosomes that will destroy foreign things that are delivered to them. And they'll destroy them because there are enzymes in the lysosomal enzymes consist of a whole bunch of different enzymes, some of which are nucleases that will chop up any DNA that it receives or RNA into individual nucleotides. Others are proteases that will, when vesicle is delivered to the lysosome, will destroy proteins into individual amino acids. Others are glycosidases that will chop up into sugars. Lipases chop up fats, phosphatases will remove the phosphates, sulfatases will remove the phosphates. Lipids are destroyed by phospholipases. So these are really enzymes of destruction. And this is showing in EM that lysosomes look like all kinds of different kinds of shape, and they're sort of look garbagey, and they are basically an amorphous kind of organelle that has all these enzymes in it. And without it, you wouldn't be able to live. Your immune system wouldn't function, and other cells have them as well, because cells need to break down things that they're getting from the outside world into individual, for nutrients, to break them down into the amino acids, amino acids and nucleotides that they can use. Does that make sense? Okay, so enzymes of destruction are in the lysosome. These are acid hydrolases, so they work best at a pH 5. And the lysosome is, we said that there was a pH gradient along the secretory pathway. Do you remember that? With the cis-Golgi being slightly more acidic than the ER. But the further you get, the more acidic it is. And the lysosome is the most acidic with a pH of 5, which is extremely acidic. So there's only like 0.10 difference, a tenth of a difference of pH unit between the ER and the cis-Golgi. So there's several, this is 5 is way different than 7. Okay, so let's go to a clicker question then, as we consider this. So any organelle can break down, and when an organelle need to sort of be broken down in order for turnover, in a cell, when lysosomes break, so when they spill their contents, why don't the lysosomal enzymes destroy the cell? So these lysosomal enzymes are, when they break, when they're turned over, the lysosomal enzymes are released into the cytosol. Why don't, and I just told you, they're lipases, they're nucleases, why don't they destroy the cell? So, and it looks like the vast majority of you picked the correct answer, which is that the enzymes only work in an acidic pH, choice D. So the enzymes might eventually be ubiquitinated and destroyed in the proteasome, but that would be too slow. They're enzymatically active. You need to instantly get rid of them. So the enzymes are reverse translocated through the translocon is not a good choice because that happens in the ER. And these other are possibilities, but they're all too slow. So the reason they immediately stop working is because the enzymes only work in an acidic pH. And if we look at this lysosome right here, these are acid hydrolases, meaning that they work, their enzymatic activity is best at pH 5. The cytosol has a pH of 7.2. So when this breaks and releases, this is a small volume versus the whole cytosol. So the cytosol isn't acidified immediately. And those will release these enzymes, and they're not going to work at pH 7.2. Their pH optima is at pH 5. How does this kept so acidic? The lysosome is loaded with proton pumps that will be pumping in hydrogen, lowering this pH. Same story with the rest of the pathway, the secretory pathway. There are proton pumps in various concentrations that will control the pH of the lumen of whatever organelle they're in. So you could predict there are the most proton pumps in the lysosome, and there will be more proton pumps on the trans-Golgi network than on the cis-Golgi network, because we know that the trans-Golgi is more acidic than the cis-Golgi, although the trans-Golgi is less acidic than the lysosome or the pre-lysosome. It's more and more acidic the more you go. Okay, questions? Okay, so those are the lysosomes then. Here they are right here, their pH optima. And then how then do we get from the trans-Golgi network to the lysosome? And it's the same story that you've heard before. So we have a signal on lysosomal enzymes that's recognized in the trans-Golgi network, and specialized vesicles will recognize that signal and bud from the trans-Golgi network to bring these lysosomes to the lysosomal enzymes to the late endosome, which will eventually become a lysosome. So we can think of this as lysosomal pathway. Once they've once they hit the late endosome, they're going to be the lysosome. And this is how it works. It's a familiar story to you all because it uses the same principles we've seen before. So the signal is a little bit different. Instead of being an amino acid sequence, the signal is mannose-6-phosphate. So it is a phosphorylated sugar that's recognized, and it's recognized by a receptor in the trans-Golgi network called the mannose-6-phosphate receptor. And this is how it works. So lysosomal enzymes, when they are put into the ER, received N-linked, so these N-linked glycosylation, which you know happens in the ER. So here we have it right here. And then once they hit the Golgi, mannose that's on the N-linked glycosylation is phosphorylated. So it's on the 6th carbon. So it's a mannose-6-phosphate signal. And this shows that happening. We have a protein, we start when the protein's exited the ER, so it's gotten the N-linked glycosylation in the ER. In the cis-Golgi, there are specialized enzymes that will recognize lysosomal enzymes and only lysosomal enzymes, so they bind to lysosomal enzymes and modify them by putting a sugar, by phosphorylating a carbon on mannose, on that N-linked sugar. And then so the protein travels through the Golgi, through cisternal maturation, and then when it is recognized in the trans-Golgi network, there are receptors here called mannose-6-phosphate receptors in the trans-Golgi network that bind to AP1 plus clathrin coats. So remember, those are coats that form on the trans-Golgi network. And they recognize that AP1 plus clathrin coats recognize this mannose-6-phosphate receptor. It will carry then those lysosomal enzymes over to late endosomes, and these will become lysosomes. So that's how we divert proteins away from the trans-Golgi network. On the other hand, regular secretory proteins that are going to be secreted don't have a signal on, don't have this mannose-6-phosphate signal. They might have N-link sugars, but it's not, those N-link sugars are not going to be modified with this mannose-6-phosphate signal. And so that signal, when it gets here, is ignored by the mannose-6-phosphate receptor, it's not bound to it. And those proteins will end up just going up to the plasma membrane. And that's why the trans-Golgi network is a sorting station. Questions? Yeah, question. What was the question? Why do we need the late endosome? Why doesn't it go directly to the, in the pathway, in the secretory pathway, it's actually really fluid, the compartments that go from place to place. So they get these names, but it isn't it's really a continuum from the late endosome to the lysosome as as these compartments get more and more acidic. So it isn't something like the nucleus or the mitochondria that we can point to and say, this is really distinct. It's really this amorphous membranes that are continuously in flux. And we have, through tradition, just called that the pre-lysosomal, late endosomal compartment. Does that make sense? Yeah, good question. Other questions? Okay, so let's go to this clicker question. You have a mutant cell that's unable to do N-link glycosylation. What would happen to the lysosomal enzymes? Shift to the correct answer, but still some incorrect ones. So hardly anyone picked lysosomal hydrolysis would be degraded. And hardly anyone picked lysosomal, I think no one picked lysosomal enzymes would be found in the cytosol, because you all recognize that this is part of the secretory pathway. And then we had a bunch that picked that they would concentrate in the trans-Golgi network, a few that picked they would concentrate in the ER, and we saw a shift to the correct answer, which is lysosomal hydrolysis would be secreted from the cell. So can someone who picked the last one explain their reason? Go ahead. You picked E first?
I picked C first.
Oh, you picked C first. Great. So you shifted in your discussion. So tell me your logic for the shift.
I choose the C also, and I pointed out that there's no particular receptor that makes the constitutive secretory pathway. So then it wouldn't get concentrated inside the trans-Golgi network. It would just be secreted.
That's correct. So the constitutive pathway, is the next slide show this? Yeah. No, the constitutive pathway, I have a slide somewhere. Yeah, the constitutive pathway going along the secretory pathway, you don't need a signal for it. You need the initial signals to get in at the ER, to get into the ER in the first place. But once you're there, you don't need a signal to go into COP2 protovesicles. You don't need a signal to go through the Golgi. You don't need a signal to go from the trans-Golgi network out to the outside. That's a constitutive pathway. So you do need a signal to go into the trans-Golgi network, and that is this mannose-6-phosphate signal. It was a complicated question because you also had to recognize that the mannose-6, yeah, go ahead. I misspoke, yeah, thank you. I realized that as soon as I was like, I did a replay of the tape in my head. You don't need a signal to go into the trans-Golgi network. You need a signal to leave the trans-Golgi network and go to the lysosome. And that signal is, or pre-lysosomal compartment or late endosome, that signal is the mannose-6-phosphate signal, which was a modification of an N-linked sugar. That modification happened in the cis-Golgi. So if you get rid of any kind of signal, once you're in the ER, so not the ER, not the ER signal sequence or the start transfer sequences, not those. But once you're in the ER, without a signal, you'll make your way to the outside world. And that's where the shift happened, where people realized that, right here, two things. One is that N-linked glycosylation, the mannose-6-phosphate, without N-linked glycosylation, you wouldn't have the mannose-6-phosphate signal. And without that signal, you don't have any signals to be bound by AP1 plus clathrin codes. So, and the sugar you failed to be secreted. Question here.
So what would have to happen for it to concentrate in the trans-Golgi network?
what would